Welcome back from break. Now we are going to start talking about the topics of chapter 15, okay? We only have 3 more chapters that we're going to cover between today and Tuesday, or at least that's my intent. And then the first one that we're going to talk about is lipids. Whenever we talk about biochemistry, it's important that we understand the components of an animal cell, okay? And because chapter 15, 16, and 17 are going to be in the final, it's important that you guys know the components of an animal cell. So let me just write a note here. Know the different components of an animal cell. So we have already talked about carbohydrates, okay, when it comes to the biomolecules in biochemistry. Because we are talking about lipids, let's understand that lipids are going to be found in different compartments of an animal cell. Whenever we're talking about an animal cell and for the upcoming chapters, let's understand that the main compartments that we are going to be discussing is the nucleus, the cytoplasm, the cell membrane, which we will be talking about today. And other than that, we will be talking about the ribosome. As you can see, we are going to learn next week how the nucleus of the cell is the control center, because in there we have a biomolecule known as the oxyribonucleic acid, and you may know it as DNA. Okay, we're going to talk about that on Tuesday. Then we have the cytoplasm, which we're going to see some biochemical pathways that occur in there in order to give rise to some nucleic acids. We have the ribosome, which is also going to be coming handy next week. For today, the one that we're mainly going to be focusing in is going to be the cell membrane, okay? Because in the cell membrane, we have actual structures of lipids that make up that area of the animal cell. And these cells are going to be the cells that we have in our bodies, okay? Now, we're going to see later in this chapter how the cell membrane is made out of these double layer, and as you can see, is out of fatty material, so lipids. I forgot one more component that is important and we will be talking about, and that is the mitochondrion, okay, or the mitochondria, if you talk about it in terms of plural. The mitochondrion is where energy reactions actually happen. That is mostly talked about in chapter 18, which we will not be discussing this summer, but understand that also lipids, yes, that's where you produce ATP. But lipids are also processed through the mitochondria, again, to generate energy inside of the cell. So again, when it comes to the final exam, know all the different components of the animal cell, but as we go and we talk about the different chapters that we're going to be covered, understand that some of these components we will be talking about in detail, just because the biomolecules that we will be discussing are going to be part of that component of an animal cell. So let's now move on and talk about lipids. So what are lipids? So as you can see, lipids are biomolecules that contain fatty acids or a steroid nucleus. So when it comes to structure, there's not one single structure that defines all of the lipids, okay? They're going to be very diverse in terms of the structure. Now, what they all share in common is going to be their solubility in organic solvents, but not water. Understand that the majority of these lipids are actually going to be organic it's going to be soluble in organic solvents and not water. So that means that the majority of the lipids are going to be non-polars. Now, when it comes to the word lipids, it's actually derived from the Greek word lipos, which means fat. And as I mentioned, when we look at the components of an animal cell, we can find these structures in the cell membranes. Today, we are going to see how some vitamins actually have these components and also in steroid hormones. Let's first look at the first class of lipids that we have inside our cells, which are known as fatty acids. So the reason why they're called fatty acids is because when we look at their structures, the acid portion comes from having a carboxylic acid in their structure. Fatty comes from the concept of having a lot of carbon and hydrogen chains. So because they're numerous, actually, the ones that we are going to be discussing are between 12 carbons to 18 carbons in length. That's what makes them fatty, because it has a large non-polar character. Now, understand that when it comes to fatty acids, they are categorized into saturated versus unsaturated. And what makes a saturated fatty acid is going to be by the presence of carbon-carbon single bonds, and those carbons are going to have hydrogens in the molecule. Now, when we have a unsaturated fatty acid, is going to contain one or more carbon-carbon double bonds in their structure. When we look here, the illustrations for fatty acids, you can see that we have several ways to illustrate them in the condensed structure, and you can also see the skeletal structure for them. Understand that when it comes to quizzes and exams, any of those are actually going to be utilized on exams and quizzes, and at this point is going to be the final exam. Now, on table 15.1 in the textbook, this table is extensive, so I actually broke it into 2 slides. We have the structures and melting points of common fatty acids, okay? It is important that out of this table, let me just put it that you need it for the final exam. The information that it is crucial for the final exam from this table is going to be everything except where we have these fatty acids, okay? So you should have this table handy because you will be asked questions based on the name, carbon atoms and double bonds, melting point, and structure for your different fatty acids. So in this slide, on slide 5, what we have is the list of saturated fatty acids that you guys need to be familiar with. And as you can see, we have lauric acid, which has 12 carbons, and there's no double bonds because all of these fatty acids are saturated. Then we have myristic acid, which has 14 carbons in length. We have palmitic acid, which has 16 carbons in length, and we have stearic acid, okay? So again, these fatty acids are going to be present in certain products, and I know that that is the 3rd column. So for example, lauric acid is the fat that is mainly found in coconut products. So whenever you consume or utilize coconut oil, lauric acid is what is present in there. If you consume nutmeg, then you have myristic acid. Palm oil is what gives rise to palmitic acid. And then, for example, if you consume lard or other animal fat, stearic acid is going to be the fatty acid present in that food substance. Table 15.5, again, this is the other half of it. So let me just include that you need this for the final exam. And again, similarly, we need name, carbon atoms and double bonds. We need the melting point. We need the structures, okay? So when it comes to the unsaturated fatty acids, understand that they're going to be divided into two categories: mono unsaturated and polyunsaturated. So if your fatty acid is considered mono unsaturated, that means that there's only one carbon-carbon double bond. If your fatty acid is actually considered polyunsaturated, that means that it has more than one carbon-carbon double bond. In the category of monounsaturated fatty acid, we can see that we have palmitoleic acid, which has one carbon-carbon double bond. We have oleic acid, okay? So as you can see in butter, okay, we can have palmitoleic acid. Out of olives, pecans, grapeseed oils, we can have oleic acid. Then we have the polyunsaturated fatty acids, which is linoleic acid, linolenic acid, and arachidonic acid, okay? So notice that even though before it was illustrated that it's between 12 and 18, we actually have fatty acids that have more than 20 carbons. We have 20, 22, 24, so on and so forth. It's just that in the book, there's a selected number given to you guys. But from the polyunsaturated fatty acid family, here we have linolenic acid, linoleic, linolenic, arachidonic acids in terms of the structure, okay? So remember, this table, 15.1, we are going to see throughout this chapter how we're going to utilize this information, okay? Now, let's understand that maybe in everyday life, you guys have heard the term omega fat or, you know, omega oil, okay? Put a 1 in the chat if you have heard of that term. Put a 2 in the chat if you know what it means. I see that a number of you have heard of the term omega. Can anybody want to share what they know about the omegas? Because I know that there is out there some knowledge of like, oh, omegas, this is good for you, or is it bad for you? So what has people heard about this omega thing about food products?
Is that the same thing that's like in the fish oil that's supposed to be really good for you?
Exactly, yes. So like the fish oil, and it was also said on the chat, yes, that they have omegas in them. And it is thought that when it comes to the omega terms, they're really good for you. And indeed, you guys, the omegas are something that is good for you, okay? I really don't know if they regulate blood clotting. I don't know that fact of it. I know that they're utilized because in the body, our cells can only make up to 1 omega or 2 omegas. The 3 omegas you need to get in your diet also as a source of energy is really good. Like your body actually spends energy trying to process these things with high omegas. Yeah, I really don't know in terms of like other effects like blood clotting, all that kind of stuff, but I know that it's really good for brain. And basically the cells, because these omegas can also be incorporated into the plasma membrane, that's good. Oh, there you go. So there's a recommendation of eating fish or obtaining this fish oils at least 3 times a week. And then you can get it as a supplement, you guys. You can get the omegas as a supplement. But let's actually define, when you're pregnant, it is said that folic acid and DHA, that is your jam. Because folic acid helps with development, and the DHA for development of the brain specifically. Again, another thing why the omegas are so important. But let's actually define what is that omega term. And the omega number is specifically given to unsaturated fatty acids, okay? Now, we learned previously when we were naming carboxylic acids, how when we had a particular carboxylic acid and we decided how to count the number of carbons in our fatty acid, we started with the carbon that was part of the carboxyl group. That you can see here that it is highlighted in pink. When we're trying to define the omega term in a particular unsaturated fatty acid, understand that the omega term or the omega number in your unsaturated fatty acid is going to be given by start counting from the CH3. And the omega number simply tells you the number of carbon-carbon double bonds that you have in your structure, okay? So one molecule that was mentioned by you guys when you unmute yourself, but also in the chat, is DHA. As you can see, DHA, which has that ginormous name, has 22 carbons in length. But when we observe, okay, the molecule itself, look, it has 1, 2, 3, 4, 5, 6 carbon-carbon double bonds, okay? So because there are 6 double bonds in the systems, that means that DHA has 6 different omega numbers, okay? So if we start counting from the CH3, one, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So when we start counting from the CH3 end, then we say, hey, DHA is going to be an omega-3, omega-6, omega-9, omega-12, omega-15, omega-18 unsaturated fatty acid. That's why when sometimes you buy certain oils and I see in the label that it says, oh, and this has omega-3, 6, 9. Sometimes people that don't have chemical knowledge assume that these are all different biomolecules. No. The omega-3, 6, 9, depending on the composition of, for example, oils, which is mostly when we see them. And it is an oil just because of their chemical composition, as we're going to see throughout this chapter. It can be referring to a single molecule. So again, DHA, what will be the classification of it? Is it unsaturated, or sorry, is it saturated fatty acid? Is it monounsaturated, or is it polyunsaturated? What will be its classification? Which one of those 3? Saturated, monounsaturated, or polyunsaturated? Excellent. Polyunsaturated, because it has more than one carbon-carbon double bonds. So when it comes to its omega number, again, it's going to refer to the first carbon in the carbon-carbon double bond that are present in the molecule. So if we go back into the table that it is provided into, our chapter, let's actually define the omega terms for, or the omega numbers for several of these unsaturated fatty acids. So to practice, I want you guys to define what is going to be the omega number for palmitoleic acid and linolenic acid. So I'm going to give you guys 2 minutes to determine the omega number or the omega term for those two unsaturated fatty acids. Excellent. I already see the answer on the chat. Great job, you guys. So when it comes to the palmitoleic acid, and we have 1, 2, 3, 4, 5, 6, 7, 8. You can see that this is an omega 7 fatty acid. When it comes to linolenic acid, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's more carbon atoms, but I'm just naming those. This is an omega 3, omega 6, omega 9 fatty acid. Any questions on the omega number or omega term for an unsaturated fatty acid? Then let's look now at specifically the structure of these unsaturated fatty acids more closely. Understand that when it comes to unsaturated fatty acids, they can be written cis and trans configurations. But let's note that when it comes to natural occurring unsaturated fatty acids, nature has most of its unsaturated fatty acids be in cis form. Trans fatty acids, there's very, very few examples in nature. Actually, the preferred conformation for an unsaturated fatty acid is the cis bond. Trans fat, it is something that was created in this industrialized food era, because we're going to see how there are certain reactions that have been done with these fatty acids and other molecules that have fatty acids, which it was where the bulk of these trans fats or trans fatty acids were created. Another thing that we should note in terms of the physical properties of fatty acids is the trends that we have in melting points. As you can see here, we are comparing two different fatty acids that both have 18 carbons in length. So on the left, we have stearic acid. which has 18 carbons, no double bonds. On the right, we have oleic acid, which still has 18 carbons, but there's one double bond. Now, we can see that there's very striking differences in the melting points of both of these structures, okay? So the melting point of stearic acid, it is 69 degrees Celsius, while the melting point of oleic acid, it is actually 14 degrees Celsius. So again, same number of carbons. The only difference structurally that these two fatty acids have is the presence of a double bond. So as I mentioned to you guys earlier, when it comes to fatty acids, we know that fatty acids and other lipids for that matter are non-polar molecules. So if they are non-polar molecules, as we learned previously, there are attractive forces that are going to be present in these molecules inherently. So if you are a non-polar molecule, let's put it on the chat. What is the strongest attractive force that you will have? So for non-polar molecules, what is the strongest attractive force? Great job, you guys. Dispersion. So what we see is that both molecules will have dispersion forces, but because their structures are different by the introduction of that double bond or the presence of that double bond in the structure, then the dispersion forces, we are not going to have them in the same degree. So the molecule on the left, the stearic acid, has more dispersion forces compared to oleic acid. One of the things that we can see in saturated fatty acids is that since there is no double bonds, these molecules can actually stack very well. So if we utilize an analogy of something that can stack very well, okay, but it is the same substance, let's imagine that the fatty acids that are saturated are going to be able to stack just like Pringle potato chips. So since the surface area in those tails, in the saturated fatty acids, okay, are able to stack, then that is the concept that allows these molecules to have higher melting points when compared to other fatty acids that have the same number of carbons, but they have double bonds. So what happens to the stacking ability of unsaturated or polyunsaturated fatty acids? Well, it is disrupted because of the carbon-carbon double bond. So the carbon-carbon double bond in the structure introduces what we know, or we call in biochemistry, a kink. So it creates a kink in the molecule. So this quote unquote kink in the molecule doesn't allow those unsaturated fatty acids to stack as well. So we can utilize the analogy that the stacking ability of my unsaturated fatty acids is going to be like Lay's potato chip. It's not that they cannot stack, it's that they cannot stack as well or as high. So utilizing that, okay, and not being able to stack well is what makes unsaturated fatty acids to have a lower melting point. Now, if we compare the values for melting points among as you can see, saturated fatty acids, the longer the carbon chain, the higher the melting point. So again, just to think about it, the longer the tail, the longer the area that you have for stacking, that means that your melting point goes up. So if you compare lauric acid to myristic acid, to palmitic acid, to stearic acid has the higher melting point, because it has more carbons in it still. It's going to stack better because it has a larger surface area. Now, when we are comparing those numbers to the unsaturated, look at the values for unsaturated. In the unsaturated, as you increase in the number of carbon-carbon double bonds, those cis bonds that we have in those structures, then your melting point is decreasing. So even though arachidonic acid has more carbons, because it has 4 double bonds, it will have a lower melting point. So understand that melting point is affected, again, due to attractive forces being diminished by the presence of these carbon-carbon double bonds in unsaturated fatty acids compared to oh, the title is wrong. Let me just fix it. Saturated versus unsaturated. Let me write the word. So when we are comparing unsaturated fatty acids versus saturated fatty acids, okay? Any questions on the trends for melting points for saturated versus unsaturated fatty acids? Then let's move on to the next concept, which is, what is one of the roles that we see for fatty acids in the body? In addition to be involved in building structures of other lipids, fatty acids can also act as hormone-like substances, okay? Because understand that some fatty acids, and specifically here, we're talking about the example of arachidonic acid, it can actually be utilized in the body to signal that the body is undergoing pain, fever, inflammation. So it is part of the inflammation response. So what happens is, and we can see in the image here, that for example, unfortunately, you know, we have this example, this person hit the thumb trying to hammer. The response, which again, you can think about it, it happens super, super fast. The response is that in the body, the arachidonic acid is actually converted into what is called a prostaglandin. And a prostaglandin has that second structure that we have here. So this is a prostaglandin. Now, the production of this prostaglandin is what continues to be processed in the body, and it signals the body, hey, we got pain here. Or in other cases, hey, we have fever, or hey, we have inflammation here, okay? So whenever you have pain, fever, inflammation as the response of the body, one of the things that you could do, as long as you're not allergic to it, is to consume non-steroidal anti-inflammatory drugs like aspirin. And what is the role of that non-steroidal anti-inflammatory drug? To block the production of prostaglandin. So the analgesics that you may consume, what is going to do is specifically stop the enzyme that takes arachidonic acid into prostaglandin, and that's how it relieves that signaling process. So as you can see, fatty acids are not only utilized in the body to produce other molecules of lipids, they can also be utilized as signaling molecules in the inflammatory system or the inflammatory response. So now let's explore other lipid structures that contain fatty acids in them. And the first one that we're going to explore are waxes, okay? So what are waxes? Waxes are going to be structures in which we have one ester bond. And that one ester bond is going to link a fatty acid, which had a carboxylic acid, and we're going to link them to long chain alcohols, okay? And when I mean long chain, these alcohols that we're pairing this with can be 14 carbons, 26 carbon, 28 carbon atoms in length. And you can see here that in general, we summarize them from 14 to 30. So why are waxes important? Understand that waxes can be produced by animals or they can be produced by plants. The main thing about waxes and why they're so important in biological systems is that they prevent loss of water. And at times they repel water. So if you want to keep the moisture in, then a wax is something that, again, is produced naturally. So for those of you that may have noticed, whenever you buy peppers, whenever you buy apples, okay, I understand that industrially, there may be some chemicals and some waxes that are put on it. But when you buy them organic or you buy them at the farmer's market, you can actually see that there is like that wax texture on those items. For those of you that are Latinos like me, like for example, whenever you're trying to make chile relleno or making anything like that, like you may have noticed, if you know how to cook or if you've seen your mom make it, I'm not trying to make anybody hungry, that people put in the flame, let's say that you're trying to make chile relleno, that all of a sudden you see that your mom, your grandma, or yourself, because you know how to make it, you burn the pepper straight on the flame, right? So that is to remove that waxiness that is already found in the chile poblano, because that's what made the chile retain its water. So what you're doing is actually burning the waxes that are already naturally there. In animal feathers, we can see that the waxes are going to be coating those feathers in order to allow, when it rains, the feathers not to get, full of water. And for example, one of the products, there's actually companies that are built utilizing waxes. So for example, if you use the products of bird bees, those products, there's a line in the products that they use that they're using beeswax. And again, these are waxes that are naturally produced by the bees, okay? Downy has wax in it too. I don't know the answer to that, because when it comes to fabric softeners, I use them. I'm a Suavitel person myself. I haven't looked at the chemical composition of fabric softener, if it's a wax or if it's a polymer. I really don't know. So when it comes to fabric softener, I don't know if it's a wax or if is, yeah, I don't know when it comes to fabric softener. But understand that, again, waxes are natural. They're produced by plants, they're produced by animals, but the main thing is that they prevent loss of water. Now, other larger lipids that also have fatty acids are triacylglycerols, okay? And triacylglycerols is how we store fat. As I mentioned to you guys in chapter 13, we store sugar in our bodies in the form of glycogen. Well, we also store fat. And the way that we store fat is in the structure of a triacylglycerol. Why is it that we store sugar and fat? Because whenever we are not consuming food, we need to make sure that our body still is energized. So the glycogen produces a short-term storage, while fat produces a long-term storage, okay? Now, how are these triacylglycerols coming together? Well, in order to make a triacylglycerol, the key is that prefix tri. Tri basically means that we have 3 fatty acids that are going to be linked to one molecule that has 3 OHs. So as you can see here, triacylglycerols are actually esters of glycerol. And when I draw the structure of glycerol, it is the following molecule. It's a CH2 bonded to OH, CH bonded to OH, CH2 bonded to OH. That's what glycerol is. So to this molecule that contains 3 OHs, we are going to link at each of those OHs a fatty acid. So you can see how the body actually can do esterification reactions, okay, to make these type of molecules, okay? Now, understand that these triacylglycerols, which are also known as triglycerides, they don't need to have the same fatty acid in it. On the molecule that we have on the left, we can see that that triacylglycerol has the same type of fatty acid in it, okay? So when we look at the structure of the molecule on the left, okay? And if we go back to the structures that we learned specifically earlier in this chapter, what type of fatty acid is present here? Saturated or unsaturated? Let's put it in the chat. What type of fatty acid is in this structure? Saturated or unsaturated? You guys picked the wrong 50. It's not unsaturated, it is saturated. How you know the difference between saturated and unsaturated is the double bond, right? There's no double bond there. Since there's no double bond, these are just saturated fatty acids. Now, which one specifically, it is shown here. If we look at the number of carbons, we have 1 carbon plus 16 carbons, that's 17 carbons, plus 1 more, that's 18 carbons in length. Now, the saturated fatty acid that has 18 carbons is stearic acid. So this molecule has 3 stearic acid molecules in it. So this comes from glycerol combining with 3 stearic acids, okay? Now, as I was mentioning, triacylglycerols or triglycerides don't need to have the same stearic acid, or sorry, the same type of fatty acid in it. You can have what is known as a mixed triacylglycerol, okay? So, as you can see here, very distinct in the molecule, if we have an unsaturated acid or an unsaturated fatty acid like oleic acid, we can see that the double bond is written, okay? So it is important for the final exam that if I give you the structure of a triacylglycerol, in other words, a triglyceride, that you will be able to look at the fatty acid portion of the molecule and identify the different types of fatty acids that are in it. Just because we understand that the portion of the molecule that comes from the carbonyl, that is from the fatty acids, okay? So in this triacylglycerol, as you can see, the first fatty acid in it is stearic acid. The second one is oleic acid. The last one, which is a 16 carbon saturated fatty acid, is palmitic acid. Questions about waxes or triacylglycerols? Let's now learn about what is the difference between a fat and an oil. Because I know that in general, people use these two words interchangeably, but It actually, they are kind of not, because a fat is something that is usually solid at room temperature, okay? Now, what defines something being called a fat is because they are coming from animal products, okay? So if this particular triacylglycerol comes from meat, milk, butter, cheese, you know, as long as is specifically from an animal product, that is considered a fat. Oils, on the other hand, are usually going to be liquid at room temperature, okay? So that is their state of matter. But what really defines something as an oil is going to be that it comes from a plant source, okay? That's why we have palm oil, coconut oil, olive oil, corn oil, safflower, avocado, which is super delicious, you guys. I don't know, anybody has tried avocado oil? It's so good. And it's actually really good for you. Like, you can use it for frying. I personally don't like frying with olive oil. For me, it has like an aftertaste that I do not enjoy. Oh yeah, and for hair is good too, like coconut. Like, I know that people do keratin, but coconut for hair and avocado oil is super good. It gives like a nice shine and moisture in the hair. I'm telling you, the proteins in your hair will appreciate them. So let's now understand that if we have fats and oils, the composition of the fatty acids in their triacylglycerol, how does it affect specifically their state of matter, meaning if it's a solid or if it's a liquid. So in the following table, okay, we have two pieces of information that is presented to us, okay? Understand that here, this table tells you what is going to be the state of matter of a particular substance, if it's solid or if it's liquid, okay? So when we compare these in terms of being solid and liquids, we can also appreciate that this bar graph can tell us if we have in a particular substance polyunsaturated, monounsaturated, and or saturated fatty acids in it. Let's observe how that information gives us an insight into the state of matter, okay? So let's look at the things that are solid, okay, naturally at room temperature. We have beef tallow. Beef tallow is going to be beef fat. We have butter, okay? And we have coconut oil, okay? We can see that those three substances happen to be solid, okay, at room temperature. Now, when we look closely at the bar graph, what they have in common is a large blue bar, or in the case of beef tallow, something that is kind of like half and half between the blue and the orange. When we look at the information of the bar graph, we can see that the majority of those products, beef tallow, coconut oil, butter, they have a high number of saturated fat, or sorry, of saturated fatty acids is the correct term, in their structure, okay? So we saw before that if a molecule has saturated fatty acids versus unsaturated fatty acids, then we're going to have a higher melting point for things that are have saturated fatty acids in it. Now we can also see a second trend, that the more carbons that we have, or I shouldn't say carbons, let me backtrack on that. The more saturated fatty acids we have in a molecule, then the consistency of it will be solid. Let me just write that in words. So these guys are solid because they have more saturated fatty acids in their structure. Okay? The ones that are liquids, these are liquids because they have more unsaturated fatty acids in their structure. So we can see two trends. The ability to identify in a particular molecule the presence of saturated or unsaturated fatty acids can tell us if something is going to be solid or if something is going to be liquid. And as you can see in the words in red, that also gives us an insight into the melting point, as we observed previously when we were comparing stearic acid versus oleic acid. As you can see, the trend is that vegetable oils have low melting points, okay? When compared to things that have more saturated fatty acids, simply by the fact that they have more unsaturated fatty acids in their structure. So understand that if you are comparing different triacylglycerols, okay, the ones that are going to be liquid are going to have more unsaturated fatty acids. The ones that are solid are going to contain more saturated fatty acids. And when it comes to melting point, the more saturated fatty acids and the longer also of that saturated fatty acid in your molecule means a higher melting point compared to, for example, triacylglycerols or triglycerides that have more unsaturated fatty acids. So understand that all these trends come together to define what is going to be the state of matter for these fats and oils, but also for trends in their melting points. Questions when it comes to the trends that we see for fats and oils, depending on the composition of their fatty acids being saturated or unsaturated. Then let's move on to talk about reactions, okay? So there are certain chemical properties that we need to investigate when it comes to specifically triacylglycerols. They are all summarized in table 15.3, okay? And what you see in table 15.3 is that the reactions that we're going to talk about in chapter 15 are not foreign to you guys. These reactions we actually mentioned already in chapter 11, and we mentioned them in chapter 14. So we are going to explore the reaction of esterification, that is from chapter 14. And we remember that esterification is going to be carboxylic acid plus alcohol. That's going to make an ester. Understand that this reaction in the body, we are going to utilize a biocatalyst that is called an enzyme. We're actually going to learn about enzymes next week. We also can have hydrogenation reactions. And in the hydrogenation, remember that your alkene utilizing hydrogen in a catalyst that is going to make an alkane. We also explore the concept of hydrolysis, specifically in chapter 14. And whenever we are hydrolyzing an ester, we're going to make a carboxylic acid and an alcohol. Lastly, saponification, which is one of the things that you guys are doing in lab this week, which when we are saponifying something, we're going to specifically take an ester and we're going to break it into the carboxylic acid salt or carboxylate salt and alcohol. So let's now explore these 4 reactions in the context of triacylglycerols, starting with esterification. So when it comes to esterification, this reaction is what allows us to make triglycerides, okay, or triacylglycerols. So what component of the molecule is the alcohol? The glycerol. What is the carboxylic acid? 3 fatty acids that are going to be linking to the OHs in the glycerol. So this is going to be your component of carboxylic acid. So in the body, we have these biomolecules called enzymes that are very efficient at their job, and they're the ones that are going to do the esterification. So notice that I'm not putting here H++ heat. H+ is our catalyst. Well, we have enzymes, and enzymes work with the temperature in our body to make these structures. The important thing that you guys need to know is that when it comes to the esterification for the formation of triglyceride, we're making 3 ester bonds. Now, triacylglycerols or triglycerides can actually undergo a series of reactions, starting with hydrogenation, okay? And hydrogenation specifically is a reaction that has been done commercially to produce food products like margarine, like shortening. You actually see that you may find like different things that are butter, or they have butter consistency, I should say. Because if you look at, I can't believe it's not butter. The reason why they don't call it butter is because it's not coming from animal. It's coming from, again, they take an oil, they hydrogenate it, and they make that product, okay? So how is this done? Understand that the hydrogenation process of these oils is partial hydrogenation. That's why, like, if you read the labels on the containers, it says, oh, partially hydrogenated oils, okay? And nowadays, you can actually see in the label that it says no trans fat, because in the partial hydrogenation process, what they're doing is that they're taking a portion of the original oil and they're going to hydrogenate it to convert it into a saturated triacylglycerol, as you can see in the bottom example. Doing partial hydrogenation now is allowing these, at the chemical level, these molecules to create now things that have a different state of matter. So by doing partial hydrogenation, you can actually make something that is a semi-solid fat, okay? Semi-solid fat from the point of view that now it is saturated. So technically it's still an oil, but it's just semi-solid because now it's saturated, okay? Now, having a product that contains more saturated fatty acids in their structure, okay, but incorporated into the triacylglycerol actually allows that product to have a higher melting point, okay? Now, one of the things that it is a side product or byproduct, meaning this is something other than your desired product is made, is a trans alkene. What does that mean? That in the hydrogenation reaction, sometimes this cis alkene turned into trans. Okay, and that is the byproduct. Now, trans fat is something that is not desired. The reason why we don't want trans fat, or we don't want to consume trans fat, is because our body doesn't have the biomolecules to turn those trans fat into cis fat so we can utilize them. Biochemically, we do not have that incorporated into our cells. So trans fats can lead to many things: accumulation, problems with cholesterols. So that's why, like, even you consume them, the body tries to do the best that they can, but it cannot handle all of them, because biochemically we cannot support certain conversions that need to happen in order for those trans fat to be processed. Okay, so that's why they're so bad for you. You can consume them, but I really, when it comes to the biochemistry of the trans fat, it is skipping my brain right now. Let me see if I can look it up really quick. Processing trans fat. If we eat, I'll look it up. I'll answer it on Monday. Well, I'm not a medical professional. I'm not a nutritionist. I'm not a dietitian. I am just a simple chemist, biochemist that is very passionate about food chemistry. These products do exist, okay? So I really don't know if it's better to eat butter or to eat these hydrogenated. I really don't have the expertise to say that. Some people avoid them, some people don't. So in terms of nutritional value, I cannot say specifically which way is better. That I haven't investigated like in detail, but by Monday I will figure out what happens to the trans fat, because again, I haven't taught this concept in my biochemistry class for a long time, and biochemically right now it's not coming to my brain how we deal with trans fats in the body. Now, 2 more reactions before I let you guys go. The first one is hydrolysis. When triacylglycerols undergo hydrolysis, okay, specifically in our body, we take triacylglycerols, and again, this is how we store fat. Whenever we are burning fat for energy in the body, what happens is that we have water molecules in our body, because our body is mostly water, and there's going to be a catalyst, again, another biomolecule that is going to specifically allow this breaking of the ester bond, and that is a lipase. Again, this is a biocatalyst. This is an enzyme. So that molecule, lipase, looks at water, looks at the triacylglycerol and says like, well, we need to burn fat for energy. Let's break those ester bonds. And as we learned already during the process of hydrolysis, we create an alcohol. So we have here 3 esters that we're going to break. We form an alcohol in the form of glycerol, and then we form 3 fatty acids. Okay. And then I think hopefully in human physiology, they explain to you what happens to these fatty acids and how they are processed through beta oxidations to create molecules that end up producing that energy substance that we call ATP. Okay. But understand that once these fatty acids are created, they're going to be processed through what is called beta oxidation, and that's what makes eventually after oxidation reduction reactions is what makes energy molecules that are called ATP. One more reaction when it comes to triacylglycerols, and that is saponification. So in this week's lab, you guys are actually learning about saponification. On OLabs, you can actually do it on OLabs, or you can watch the pre-lab video. We have a virtual lab in which you're going to learn how coconut oil, we can convert it into coconut oil soap, okay? In this process, what we're doing is that we're taking that fat or oil, okay? In the case of coconut oil, it is an oil. And then we're mixing it with strong base and we're boiling it. And overall, as we learned before, in chapter 14, whenever we're doing, chapter 14. So when we are saponifying an ester, we make an alcohol and we make a carboxylate salt. Okay? And that carboxylate salt is what makes the soap. So for one molecule of fat or oil, you can see that we can make 3 molecules of soap, okay? Now, when it comes to the structure of soap, we can see that there is a portion of the molecule that is ionic, okay? So technically, when we look at the structure of soap, we typically draw them as a circle with one tail, in which if I label this circle, which is the polar side, is going to be this ionic portion of it. Then the tail in my soap is going to be the non-polar side, okay? So if I label this portion is polar, and this portion is non-polar. When we come into terms that we actually explore in chapter 9, the polar portion of my soap is going to be hydrophilic. It's in love with water. But the non-polar, because it wants nothing to do with water, yeah, the Krebs cycle is how we produce energy. Followed by oxidative phosphorylation. When it comes to non-polar side, this is going to be hydrophobic. So it's afraid of water. So how is it that a soap actually cleans a surface? Well, understand that when we have soap molecules, you can see them, okay, being soluble in water because it is a carboxylate salt. It is an ionic compound. So even though it has those tails, since it is a full cation and a full anion, because we made an ionic compound, detergents are actually soluble in water. Now, understand that let's say that you have a surface, it could be clothes, it could be the counter, that it's soiled, okay? Let's say that there's grease in it. Now, understand that the role of this soap is that the tail, okay, of the detergent, because it is non-polar, and we already learned that grease, lipids, all of that is non-polar, is going to interact with the tail of your soap. Now, if you are doing laundry, you know that your laundry machine has to move. That creates, mechanical force, mechanical energy in it, because we are going to agitate. This agitation is what's actually going to allow more of these soap molecules to be interacting more with, let's say, grease, if it was what affected, the surface that you're trying to clean. So as that process continues, you guys can actually see in that fourth image that what we create is these molecules that are now soluble in water, and this is actually called a micelle. These micelles, when we look at them closely, in the middle of it is where you see the grease. because it is interacting with the non-polar portion of your soap. The polar portion, which is the circle, is what's interacting with the water, and that's what makes all of that grease, all of that dirt, whatever you're trying to clean up the surface, be water soluble. So when you're washing, let's say laundry, then when you have the rinse cycle, that's what you're hoping happen in the first half of it. Okay, that in that first process, you created enough of these micelles to clear whatever soiled the surface of your clothes. Okay, so that's an interesting question. The bubbles trap the oils in it. So the bubbles actually are a result of the molecules that are extra. Okay, so lather, which is the bubbling, which is another thing that you guys explore in this week's lab, is going to be the foaming ability that is formed by the soaps. Now, that is in a way trapped air in these molecules, and the more molecules you have, then you have a better ability to create them. Now, as you guys learn doing in this experiment, depending on the composition of your water, the foam is less available. And if you have, for example, hard water, then all of a sudden your soap is actually not as available. So I always think that when it comes to the bubbles and I'm cleaning something, I make sure that I have a nice foam, because that lets me know that I basically saturated a lot my clothes, so I will have enough micelles to clean. Okay, that's why sometimes I see the commercials for the you guys know the little containers that come in plastic, and they have detergent, and they're like they throw 2 of them, and they're like, this is gonna wash a whole load of yeah, those things. So sometimes I feel bad. I'm like, really? Like that little amount of detergent is going to clean all of the clothes? Like, I don't know. I don't use them. Oh, please. Yeah, there's people that eat them. Please don't eat them. That is detergent. That's going to make you sick, you guys. Please don't. But understand that when it comes to foaming, it is actually a good indication if you have enough detergent in it. And you will be able to, you know, clean as much soil. Because sometimes I remember back in the day when I live in apartments and I will go to the laundromat, sometimes I will see people wash clothes and then the water looks kind of gray and there's no foaming. So I would be like, is that really cleaning the clothes, or is just the clothes washing in dirty water? I don't know. But anyways, make sure that you guys have, you know, enough foam when you're doing your laundry. Now, this is all what we have for today.